I would like to thank the organizers for giving me the opportunity to present this work entitled Inhibiting Bromodomain and Extraterminal Domain Proteins as a Therapeutic Approach for NF2. Neurofibromatosis type 2 is a multiple neoplasia disorder characterized by development of bilateral vestibular schwannomas, and it is inherited in an autosomal dominant fashion, attributed to the loss of heterozygosity of the NF2 gene, which encodes for a tumor suppressor protein called Merlin. Merlin is an essential mediator in various regulatory signaling pathways, including the HIPOA pathway. One of the mechanisms through which YAP regulates transcription involves interaction with the bromodomain and extraterminal domain proteins. The BET proteins are a protein family comprised of 4 members, including BRD2, 3, 4, and BRDT, which play a role in regulating RNA polymerase II transcription. Several studies have shown that BET proteins are upregulated in different types of cancer, and consequently attempts have been made to synthesize inhibitors of these proteins. The use of BET inhibitors as a therapy for treating NF2 schwannoma has not been previously explored. Therefore, to assess the requirement of BET protein activity in the absence of NF2, we used human-derived Schwann cells that were NF2 deficient and treated them with increasing concentrations of JQ1, a BET protein inhibitor, resulting in a dose-dependent inhibition of cell growth over time. Also, loss-of-function experiments using siRNA knockdown against each individual protein identified BRD4 and, to a lesser extent, BRD2 as the primary mediators of JQ1 effects on cell growth. We also assessed the impact of BET inhibition on tumorigenesis in vivo, using an orthotopic model based on SC4 luciferase cells. Tracking the bioluminescence image signaling in JQ1 treated animals suggests that treatment partially impairs tumor growth compared to the control group. Also, comparison of the tumor weights demonstrated a significant lower average tumor weight in the experimental control group. Furthermore, immunohistochemistry demonstrated elevated levels and activity of BRD4 in human vestibular schwannomas compared to normal nerves. To determine how JQ1 inhibition impacts the NF2 null Schwann cells, we treated the cells with JQ1 or control and analyzed gene expression using RNA-seq and gene-set enrichment analysis. The gene targets that appear to mediate JQ1 effects on Schwann cells are involved in the PI3 kinase AKT pathway. We used phosphokinase array, Western blot analysis experiments, and PI3 kinase inhibitors, which all suggested that the effects of the JQ1 treatment are mediated, at least in part, via inhibition of the PI3 kinase AKT signaling. Overall, these findings implicate BET inhibition as a potential therapeutic option for the treatment of NF2-related schwannomas.